For those on Zoom, can you guys hear me?
Yeah, it's working.
Okay, perfect. Um, the Zoom link is on the link tree. All right, I guess I can begin. Um, Thank you all for coming here today. I know the beginning of this meeting was a little chaotic, at least for me. Um, but yeah, I just wanted to do a quick presentation on the advancements in precision medicine, leveraging machine learning for clinical research. Um, there we go. So a little bit about me. My name is Sahiti, and I am a third-year senior studying biomedical informatics undergrad. And I'm also in the accelerated programs to get a master's in bioinformatics by next year. I'm also a research assistant at the Jenga Research Lab, where my main expertise is next-generation sequencing analysis. Um, and so here's just a quick agenda for today. So the first half, I'll be going over a paper. It's called Machine Learning for Precision Medicine, and it's a really great introductory paper if you're getting interested into precision medicine and machine learning and how you can implement machine learning for those, I guess, applications. And then we'll do a quick break, and then I'll be going over an application with one of my projects, just so you guys can get a good idea of like a real life scenario of what the paper was talking about. So again, the paper we'll be discussing is called Machine Learning for Precision Medicine, and this was published in 2020. This is a mini review paper. And again, was just a really great introduction to machine learning and precision medicine. So without further ado, what is precision medicine? So most medical treatments are designed for the average patient. So they're focusing on one size fits all. And sure, this is mostly used, but it's important to note that it may be successful for some patients, but not others. Um, so this is where precision medicine comes into play. And the goal here is to target the right treatments to the right patients at the right time. Sorry, one second. Okay. And so another goal of precision medicine is being able to tailor prevent disease prevention and treatment based on patient individual data. And this data could include things like medical diagnostics, clinical phenotype, biological investigation, such as laboratory studies and imaging, and then also environmental, demographic, and lifestyle factors. And so all of these taken together is called multi-module data, since they represent information from multiple domains. So precision medicine has been has been evolving over the years. And this is mainly because there's been an exponential increase in the amount of biological data collected for each individual patient. And if you see the figure down here, it's just showing that exponential growth throughout the years and the data growth. And the y-axis is zettabytes. And so no, that's a lot of zeros. So we're dealing with a lot of data now. And another thing is that the field of computer science is also reaching or experiencing an evolution. Now computers are able to store more data and able to analyze large volumes of data. So the two developments go really hand in hand with computer science methodologies making use of large volumes of data collected in the healthcare system. And this really allows for the advancement of precision medicine in diagnostics and therapeutics. So I thought this was a really fun infographic, and this is just showing how genomics has evolved over the time. So we first started off with not even knowing what DNA was. So I believe it was in, yes, 1869. That's when the DNA was discovered. And from there, we were able to kind of understand the structure of it with Watson and Crick and Rosalind. And then now we're kind of at the stage where we're like, oh, great, like DNA is such an important thing, and we can actually make use of it in the medical standpoint. You know, like DNA, you can kind of look at what is regulating these factors within the cell, how you can make use of that for therapeutics, and I guess distinguishing between healthy and disease, or just between people in general. And so again, so genomics has played a significant role in the launch of precision medicine. So then again, just trying to emphasize this. So the initial focus of precision medicine was heavily focused on genomics, and this was mainly due to the fact that there were significant decreases in the cost and time to conduct genetic testing. And so the graphic you're seeing below is the cost per human genome over the years. And I did want to note that the first genome was sequenced in 2001 and took almost a decade to even get that up. But today, that cost and the time of doing this has decreased significantly, where now you can sequence an entire genome in a matter of a day or two, and it only costs about $1,000. And to this day, that cost is starting to decrease. And so now it's much more easily available to sequence the genomes. Another thing that's playing a major role in precision medicine is the omics revolution. And this basically is focusing, well, previously we were focusing on genomics and how precision medicine was making use of that. But nowadays, researchers have noticed that, oh, it's not just DNA that's important. There's RNA, there's proteins, there's imaging, epigenomics. And so you can actually extract data from all of these omics and use that information for diagnostics. And so the processing and extracting information from these complex data sets is very difficult and even impossible without the help of data science and increasing computational power and technology. And so this is where machine learning comes into play. And so machine learning is a branch of artificial intelligence where computer-based models are created to identify and learn patterns in high-dimensional data to create predictions. And so machine learning can be further subdivided into two categories. So there's supervised learning algorithms and there's unsupervised learning algorithms. So first, going into supervised learning. So this aims to identify patterns using labeled data. And so there is some known knowledge. So for example, if I were to tell you, oh, this is a dog, this is a dog, this is a cat, and I'm telling you that that's a dog and a cat, right? But what if there's now an unidentified picture here? And so the main goal here is trying to use that existing knowledge and those labels to determine what this unknown picture is. And so that's kind of the basis around supervised learning. And so some supervised learning modules are support vector machine, random forest, decision trees, naive Bayes, linear regression, and deep neural networks. I'm not going to be going into how these algorithms work and such, but I hope if you guys are interested, there's plenty of resources online to go through them. There's also unsupervised learning, where now the goal is to uncover patterns in unlabeled data. And this is very helpful to answer questions such as, are there different types of diseases? How much does a patient with a disease differ from that who is healthy? And here there's different unsupervised learning algorithms such as hierarchical clustering, k-nearest neighbors, Gaussian mixture model, and principal component analysis. So just a quick exercise. So we have two pictures here. I know we know what those are, but pretend we don't. So what are some characteristics that you can identify that distinguish these two pictures? And I'm going to open up the floor to you guys. Color, okay. Anything else? What was that? Yeah, one has a beak, the other one doesn't. Yeah. Anything else? Yeah. And like, for example, one has feathers, that one other one has hair, fur. Yeah, and so that's great because we gave in our machine learning model some pictures. It has no idea what those labels are, but it's able to distinguish what, for example, the first picture is from the second based off of these characteristics that we talked about. One has a beak, one doesn't. One has 4 legs, the other one has 2. One is made of feathers, the other one is made of fur. And so if you give this model another image, such as the one on the right, it should be able to, I guess, label what that is. So in the previous slide, we were looking at a bird and a dog, right? And so the main idea for this machine model is to categorize that. And so if you were to input this picture, it should understand that this is very similar to the bird in the previous slide. So they should both be birds. So that's kind of the idea of unsupervised learning. And so using these machine learning algorithms, there's some really important things to consider before you start implementing your data in this application. So the first one is that your input data should be of high quality with small artifacts or noise levels. The second is that the correctness of the ground truth labels must be ensured during data curation. This is because having wrong labels in your data, of course, can downgrade the performance considerably. You also want to make sure your data is as complete as possible. Now, I know missing values is kind of inevitable in some cases, and there is data augmentation methods such as randomly putting in variables in those missing cells, or taking the mean of all the values in the row and just inputting that for the missing values. But it's important to note that while you can do this, it doesn't usually lead up to the same results as just or performance compared to using a complete data set. The fourth thing is that larger data sets are generally preferred because this enables the machine learning model to learn the true variation in the data. And we don't want to risk few outliers that will affect the model negatively. The fifth thing to consider is that models should be optimized considering both precision and accuracy. And every machine learning model should be validated prior to using it for computer-aided diagnostics in support to clinical treatment decision-making. All right, so here are some advantages of machine learning that the paper went through. The first one is that most machine learning models don't have strict assumptions about the data distribution, so your data is not required to be in this normal distribution. Which is really great. And then most machine learning models can easily combine multi-omics data. And another great thing is that this data can be binary, it could be categorical, discrete, continuous. And so this really avoids the need for major pre-processing before inputting that data into the machine learning model. And then the third thing is that the more complex machine learning models can also identify multifaceted nonlinear patterns in the training data, which may not be obvious to the human eye or simple linear models. Now, the thing about having a very complex data set can also have some negative impacts. So for example, the more complex a model is, the higher chance of overfitting. And so overfitting is an idea that where the model fits too closely to the training data. And this results in the model that can't make accurate predictions in the testing data. And this is because it just adapted so well to the training data to the point that it's I guess in this case, it's stratifying the data too perfectly, like unrealistically perfectly. to the point that if you given testing data, it's not able to perform that well. And then another issue that machine learning models can run into is underfitting, where it just isn't able to capture the patterns in the data at all, as seen in this figure. And so you kind of want to find that sweet spot. All right, so some applications of machine learning in healthcare and in clinical settings is that you can combine the multi-omics data that we were talking about earlier for disease diagnosis, prognosis, and treatment. And so one example of this is, for example, predicting time until Alzheimer's disease diagnosis using genotype information. You can use neuroimaging and clinical data, such as neuropsychological measures. And by implementing all three of these data sets, you can potentially predict the disease diagnosis. So I wanted to open the floor again. And are there any applications that you guys can think of?
Finding tumors in imaging data.
Okay. Yep.
Prediction.
Any others from this side of the room? It could also be used in health informatics, where you have this electronic health records. You have a lot of data with that. And so you can use patient information about, for instance, let's say, their age, their female, male, I guess what drugs are they using? Do they have any allergies? You can use that information as well for machine learning models to predict potentially any disease, if they're more likely to get a disease or not. So yeah, and so the key takeaway from this mini review paper was that precision medicine is a novel approach to research and a clinical practice that utilizes data from multiple sources to understand and treat human disease. And machine learning is a very important component of the analysis and processing of the multi-omic data that allow for classification and prediction of outcomes for individuals and populations. So do we have any questions at this point? All right, well, I guess we can take a little break, and then I'll be going over a project that I'm doing. There is pizza in the back if you guys would like to get a slice. So I wanted to share a application of using machine learning for disease diagnostics or clinical research. And so this is a project that I'm doing right now. Before I get into it, I want to give a little bit of background. So my project is focused on diabetic retinopathy, and this is a progressive eye disease that is the leading cause of vision impairment and blindness, and it also occurs when diabetes damages the blood vessels in the retina. So this is the retina of the eye. It's this tissue in the back of it, and this is the light-sensitive tissue. And so there are 4 stages of diabetic retinopathy. There's mild non-proliferative DR, also known as NPDR, and this is characterized by the balloon-like swelling in certain areas of the blood vessels in the retina called microaneurysms. There's also moderate NPDR, where the damage to some of these blood vessels in the retina causes leakage of blood and fluid in the retina tissue. There's severe NPDR, where now you're seeing more blood vessels that are being damaged and blocked, and there's even more leakage of the blood and fluid into the retina, resulting in a much greater impact on vision. And then finally, there's proliferative DR, also known as PDR, and this is the most advanced stage of DR. There is extensive damage to the eye's blood vessels, and there's worsening circulation inside the eye. The retina can grow more blood vessels, but they tend to have abnormal formation and can cause severe damage. So this is just a good figure that's kind of illustrating the differences between a normal retina and a diabetic retinopathy retina. And so you're seeing a lot more of these abnormalities, and this causes the loss of vision that you're seeing here. And it can progress throughout the stages. Another piece of background is just a quick introduction to circulating angiogenic cells, or CACs. And so these function as a circulating reporter cell in the retinal vascular health due to its striking similarity with endothelial cells. Endothelial cells are the cells that you're seeing in the blood vessels, and they make up that lining. And so these CACs have the ability to repair endothelial injuries. And so when an individual gets diagnosed with diabetes, it leads to a phenotypic switch in the CAC function. And instead of repairing endothelial injuries, it actually has the opposite reaction, where now it's shifting to a pro-inflammatory phenotype. And so this project is in collaboration with the IU School of Medicine, Department of Ophthalmology. It's with, so we're working with Dr. Akshay Bhagwadekar. He is a professor at the Indiana School of University, and we're working with his lab in the Eugene and Maryland Glick Eye Institute. And so the objective of this project is to build a prognostic algorithm to predict the severity of diabetic retinopathy in patients. The data was taken from blood samples in their patients, and these patients have different stages of diabetic retinopathy. I also wanted to mention this is a 5-year project, and I actually joined it early last semester. So it's still, the results I'll be showing is still very preliminary, since we're still trying to get more results from these patients. And so going back to the data, so the CAC cells were isolated from these blood samples, and we're using both RNA-seq data and surface marker expression data. And these patients that we're using come from 6 different diagnostic stages. So we have control or healthy patients. There's patients with diabetes, but no diabetic retinopathy. There's mild NPDR, moderate NPDR, severe, and then there's proliferative diabetic retinopathy. And so a little bit more information about the surface marker data that we're using. So if you don't know, surface marker data is basically assessing the changes in the surface marker expression in those CACs. And this data is generated from flow cytometry. And this is a laser-based technique used to detect and analyze the chemical and physical characteristics of cells or particles. And so we're working with 10 different features, and this expression data is represented in counts. We're also working with RNA-seq data that was also extracted from the CACs, and we go through the RNA-seq pipeline. So you get the FASTQ files after sequencing. You do quality control to see if there's any bad reads, any adapters, and you trim all that out. So it's just basic pre-processing, and then you align it to a reference genome, and then you do quantification. So in quantification, you have the genes, and you're just trying to see how many of these genes are being expressed in each of the samples. And so you'll also get count data from there. You'll also do differential expression and functional enrichment, but for the purpose of this project, we didn't do those 2 steps. Instead of differential expression, we did principal component analysis, which is just a way of reducing the number of dimensions in the data set, because there's so many samples, there's so many genes, and so your data is just not in a linear format, it's in multiple dimensions. And so when you kind of bring it in into something that's more human readable, you're able to kind of distinguish, oh, these genes are showing more variability within the different samples, and that's how you kind of extract a significant genes for machine learning. Some of the machine learning models that we were using is random forest, k-nearest neighbors, decision tree, and support vector machines. The screenshot you're seeing here on the right is what was put in the grant. It's kind of showing the performance of these classifiers when applied to a benchmarking data set, and that's why we decided to use these for this current project. I will quickly be switching over to some of the results that I've gotten. Give me a quick moment. All right. So these are just some examples from some of the updates that I've given in the past to my collaborators. Let me just hide this. Okay, so after you get your data, one of the important things is doing feature selection. So you don't want to just input all of your data into the, sorry, 1 second. For those online, are you guys still able to see my screen? Is someone
It's a
online?
little hard to see.
Okay, that's what I thought. I don't know why it did that. Let me just try stop sharing it and doing it again. Oh, there we go. Are you able to see it now?
Yes.
Okay, great. Let me hide this. Okay. So yeah, as I mentioned before, this is I'll just be going through some of the results that I've shared with my collaborators. And so one of the first okay, so I'm so sorry. I don't know why that's happening. It's like cutting off in the online video chat, but okay, hopefully that should work. And so yeah, one of the first steps is feature selection. You want to remove some of those features or like those genes or the surface marker data that aren't really showing good variability between the different diagnoses. And so with the surface marker expression, the first step is to just visualize the data. How is it looking like? Again, this is very preliminary and we're still trying to get data from patients. And so we only have 5 control samples, 4 samples from those with diabetes, but no diabetic retinopathy. And then we had different stages of NPDR, but we just grouped them. So we have 10 samples of that. And then we have 5 samples from PDR. Again, I know this is very small number of samples per each category. Just wanted to mention again that we are hoping to get more samples. We actually do have more samples right now. I need to run the analysis. But yeah, as I was mentioning before, the first step is to visualize the data, seeing how the distribution is looking like. The y-axis is the count, so how many of these surface markers are we seeing in each of the diagnosis. From there, we did a Kruskal-Wallis non-parametric test to see if the means between these distributions are actually significantly different. And so from doing this, we were able to see that the TLR8 plus and CD34 plus cells, so these are cells that are containing these 2 receptors, are actually significant. From there, we did an ad hoc test or Conover test. What this does, it is telling between what 2 groups are actually significant. So for example, is the surface marker expression from NPDR and control significant? Is it between PDR and control? So for these 2 features, we saw that PDR and control was significant and PDR and diabetes was significant, and so on and so forth for the TLR8 positive and CD34 positive. We also did some linear and nonlinear modeling to help with the feature selection. So first we did linear regression using all of the features, and it wasn't that significant. And then we did it with the features that were specific to receptors and markers. And now we're seeing some better results, but still not the best since the p-value is greater than 0.05. We did nonlinear modeling with all of the features, again, not great. But then we did nonlinear modeling with just the receptor and marker features, and we were able to see that some of the features were significant. When I mean features, I mean these are like the surface marker proteins. So these two features were what were used in the machine learning model. Yeah, we can skip over that. We also did PCA analysis for the expression data. I'm not gonna go too in depth into that, but when you have RNA-seq data, there's just so many genes that you'll get outputted from that when you do quantification. And so it's really just understanding what genes are actually of use for machine learning. So in that case, you would, for instance, remove all the genes that are, giving out zero counts, for example, because you don't really need that, or genes that aren't really showing much variance between the different disease progression levels. So that's what PCA analysis does, and from that I was able to extract. I guess I can show this. Move this. So this is how a data set looks like after you do principal component analysis. These are the principal components, and there's a couple methods on how to choose how many principal components you want to use. But principal components basically just tell you how much variability do you want to consider in the data set. Variability meaning how different should the expression be between each of the diagnoses. So I chose 4, and you can extract the different genes that are being implemented in the principal components. It seems a little over the head, but just know that it's just another way of measuring if the genes are actually being significant. From there, we were able to get both the significant features from the surface marker expression data and the significant features from the RNA-seq data, and you kind of just combine it. And so this was another update that I gave in my lab. And it's kind of just showing the machine learning results that I got. And so these were the number of samples that I had. For RNA-seq, it is much harder to get that data because you have to go through sequencing, you need to put more money into it, whereas surface marker data is a lot quicker. And so we ran the different classifiers, and again, you're not really seeing great results. And this is just because there's very less number of samples. We are planning on getting a lot more, like I mentioned before. But this is just us doing that. This is again just more information about the PCA that I did. And I did do machine learning on just the surface marker expression data. And so again, we're seeing less percentages, but because there's more surface marker data than there's RNA-seq data, the machine learning models were able to give a little less biased results. But yeah, this is where the project is at right now. I just wanted to give a real world application. Again, it's still very preliminary, but I hope that gave a good understanding of how machine learning can be implemented. Do we have any more questions? Yeah, what's up?
I'm actually running into similar issue with the time. What's your proposal to for your approach to increase your significance or who is overfitting when you're running your models? Because as what I heard was it's good for really balance and large simplified. So in this case, how do you overcome?
So I think in this case, so like this isn't really the final results. And again, I mean, right now we have, I think like 15 samples per diagnosis, but that's still very less. And we're still getting more data. But I think to answer your question, I believe there are machine learning models that can handle less data. I don't remember what the algorithm is on the top of my head, but there is that. I think another thing is just you don't want to. So I actually did a project I think my sophomore year, and I was trying to predict how many Netflix TV shows will come out in 2024 based off of previous data. And so I was trying to make my machine learning model so like very complex so that it can actually fit the data. And what I noticed is that when I put in a future data, like it just went skyrocketed. And this is like, that doesn't make any sense, right? And so you just need to make sure that your model is, I guess it's okay if there's some errors that it can handle, but you just don't want to make it too perfect to the point that it's overfitting the data. Yeah, so that also comes into, taking those significant features. So I don't know, for example, if you're working with genomic or transcriptomic data, like how many genes are you actually inputting into the model? Are they actually showing a good like variance between the different treatments and stuff like that? Yeah, hope that helped. Any other questions? All right. Well, I guess it concludes the Journal 2 event. I did want to mention that, yeah, so this is the attendance form. If you haven't completed that yet, it would be really great if you can. I can expand on that.
But this is the only way people can get attendance because the other QR code was.
Oh, really? Okay. Well, thank you guys for coming today on a Friday. I hope you were able to learn something. And yeah, I guess that would conclude our event. Thank you.